Enzymes. What do you know about enzymes? Anything about enzymes you know?
They speed up reactions without being used up.
They speed up the reaction without
Being used up.
being used up. Okay, so that is a definition of enzymes. And when we are talking about biological enzymes, what we will put it as they are the proteins that function as biological catalyst, right? And catalysts are the
Mm-hmm.
that are speeding up a chemical reaction, as you said. Okay, so this is the definition for enzymes when we are using it in biological terms. Okay, now under
Mm-hmm.
enzymes we have different topics. We'll come to one of them that is known as the lock and key hypothesis. Okay, one
Mm-hmm.
check if I have a diagrammatic representation for you. One moment. Okay. Not finding it one. Okay, fine. We're talking about enzymes, right?
Mm-hmm.
Now, what is happening with enzymes? We saw that they are the protein that are breaking down, right? The speeding up, right?
Mm-hmm.
What exactly is happening? See that with the help of a lock and key theory. Now, in a lock and key theory, we have an and we have a substrate. Okay, I have found one. I'm just uploading for you. So that will give you a very clear product of what we are talking about. Here you go. Okay, this is the lock and key method. Okay, lock and key theory actually. Now, what is happening here? Here we have the enzyme. Okay, now this enzyme has something called as the active site. That is just the border surface of the enzyme. Okay,
Mm-hmm.
what is this? Is enzymes? They're all folded into 3D complex shapes. Okay, and they have these places where the molecules are going to fit, which is known as the active site, and this is the substrate. Now, in this lock and key theory or the hypothesis, the shape of the active site should match shape of the substrate molecules.
Mm-hmm.
The shape of this active site match the shape of the substrate molecules. If you see here, only if they are matching, this enzyme will help the substrate to come and bind with it, right?
Yeah.
Right? Now, if suppose there is a substrate, okay, which is square in shape, will it come and get attached to the active site over here?
No.
No, right? It should have a matching substrate, something which is matching the shape of the active site. Now, this is why, because enzymes, they are known to be highly specific molecules. Okay, enzymes are highly specific. Now, so what happens is the substrate comes and attaches to the active site. Then what we form is the enzyme substrate complex.
Mm-hmm.
You understanding what is happening? What we form here is the enzyme substrate complex. And once the loop is over here, right? They fit between the enzymes, the substrate is the lock and key. So now if this is the lock and this is the key part, they fit in it. Once after it, what happens is the enzyme, it is unchained and they release the substrates. That means the products are now released. The substrate has now transformed itself into a product.
Yeah.
Okay? You understand this concept?
Mm-hmm.
Okay. Now, the same thing when we come down and see here. We have an enzyme. We have two forms of substrate. Okay? What happens? There, if we divide this active site, this will come and fit over here, right?
Mm-hmm.
And the other part is going to fit over. That is where we get this enzyme substrate complex here. After getting attached, that means when they form the substrate enzyme complex, the enzyme again releases the product.
Yeah.
Got it? You
Mm-hmm.
understood what is lock and key theory?
Yeah.
Do you want me to write it also, or is this diagrammatic representation is enough for you?
This is enough for me.
Okay. Tell me once you're done and then.
Yeah. I wrote it.
Done?
Yeah.
Okay. So this was a lock and key hypothesis. Now we'll see what is denaturing of enzyme means. Okay, so we are done with lock and key hypothesis. Okay, then we come to denaturing enzymes. What do you understand by the word denaturing?
Um, when you can't use the enzyme anymore because it's like broken or it can't function.
Okay. Now, whenever we are carrying out a reaction, okay, whenever there is a biological reaction happening, there are some factors which we have to see. That is temperature, concentration, and I think the pH level. Okay. Now, these
Mm-hmm.
three factors will work for the enzymes. Now, if suppose there is an enzyme and which we have exposed it to very extreme high level of pH, okay? Just imagine we have put it to a very high temperature. What might happen? They are going to
Denature.
denature. Okay. Now, when we're talking about the denaturing of enzymes, what is happening is my active site is being disturbed. Okay, the shape of this active site. This is my enzyme, this is my substrate which is ready to come and bind. But because of this high temperature, what might happen is
It'll lose its shape.
it's losing its shape. So the substrate is remaining where to fit here. Okay, so what happens in this case is the enzymes or the substrates will no longer come and fit into the enzyme. And then we can say that they have been denatured. You understood what is denaturing of enzymes? This is just an overall view of denaturing enzymes.
Yeah. I've written it down.
Done?
Mm-hmm.
Okay. Now I just told you there are different factors which is going to affect the temperature enzymes, right? Now we are going to see one by one what is going to happen here. Now, whenever we're talking about the enzyme, the rate of enzyme catalyzed reaction, they all depend upon the substrate which is being used up. Substrate whether it is used up, okay, and
Yeah.
how are we going to calculate this? With the help of a formula that says rate of reaction is equal to the amount of substrate used or amount of product formed divided by the time taken. Okay, this is a formula for getting us the rate of reaction. Fine. Now we are going to see how these factors are affecting the rate of reaction. First one we are going to see about the temperature. Okay. Now,
Mm-hmm.
very simple. In simple terms, you're cooking something. Okay. Just imagine you're going to prepare a dal.
Yeah.
Simple dal you're going to prepare. We know in what temperature we are going to cook it. If it is a high flame, right? Whether I
Mm-hmm.
need to boil it into a high flame or I have to boil it into a low flame. Now, what will happen if I just keep it for a high flame and then I move away from that place? What will you observe?
Um,
What are the
it,
changes? Okay, what are the changes you will see?
it will cook slower.
It will get.
It will cook slower.
No, no, I'm telling about high flame. If I just keep a dal in a vessel, keep it in a very high flame, and I move away from that place,
Oh, will it burn?
it will get burned up, right? Similar
Yeah.
thing is happening over here enzymes. When I'm going to increase my temperature, my rate of reaction is also increasing. And what did we see? When it is increased to a very high level, what ultimately it is going to denature,
In nature.
right? Simple concept. So we have to maintain a perfect temperature whenever we are using enzymes as a biological catalyst. Okay, and whenever we are talking about rate of reaction, all my rate of reaction has to be expressed in forms of graph. So here will be the rate of enzyme, here will be the temperature, and if I need to plot a graph for this will be okay. This peak level is known as the,
Optimum.
yes, it's the optimum temperature. Optimum temperature is the best or the appropriate temperature for which the enzyme will work perfectly. So after that, what is going to happen is just going to slow down.
Yeah.
That means it is getting denatured. When we come to this point, that means the enzyme active site is already denatured.
Yeah.
Okay,
Mm-hmm.
done with temperature. Shall I move on?
Yeah.
Second, we are going to see is the effect of pH. Okay, why do we use pH actually? Why are we using pH values?
To see how alkaline or acidic something is.
Yes, to check whether any solution, any kind of reaction is coming under an alkaline or an acid. Okay. Now again pH. Whenever we are increasing the pH or we are decreasing the pH, it is going to affect the rate of reaction. Okay, that means each enzyme works best at specific pH value. Okay, each enzyme has
Yeah.
its own specific value, and now the optimum pH value for any enzyme work is 7.5. What is 7.5? In which category will it come? Either acid or a base?
It's like neutral, but more on the base side.
Okay, somewhat near to the neutral side, right?
Yeah.
Okay, so 7.5, say 7.5 to 8. Okay, That is the optimum pH. Now again, to represent them in the form of graph. Here will be the rate of enzyme, pH, okay, and this will be 8, pH 8. Okay, this is my optimum pH. So when it is coming below, I mean more than 8, okay,
Uh-huh.
when it is moving toward the base side, what it is going to happen, it is going to denature itself.
Yeah.
Okay.
Yeah.
Third factor we are going to see is about the substrate concentration. Okay, the concentration part. Now, what is going to happen? Enzymes, again, they will work best if there is a plenty of a substrate, right?
Mm-hmm.
I had showed you a pictorial representation where I have this enzyme. I have a substrate which is this way, right? Basically, they both are going to fit. If it is in a spherical form, it will fit directly. But I have two different forms, right? Anyways, these
Yeah.
different forms are going to fit. Just imagine if one of them is not there. Will it go and bind to the active site?
Mm-hmm.
Will it
So
bind? No, right? There should
yeah.
be plenty of substrate available for the enzymes. So as the concentration of the substrate increases, what is happening? Rate of reaction is also increases. Okay, but whenever we talk about the substrate concentration, my rate of enzyme is not going to increase forever. Okay, now there will be a point where this enzyme becomes saturated and there is no more substrate which can fit. Now same case, I have few more substrates, right? Okay, few more substrates, semicircle ones, which are available here. We said that we need us plenty of substrate. So now these two substrates have come and they have attached themselves here.
Yeah.
Okay, these two ones have attached themselves, and there are still plenty of substrates available. So do you think they will come and forcefully enter this active site?
No.
No. Why? Why? Because my enzyme has already accepted a substrate and now it has reached a saturation point where it is like, okay, fine, I don't want any more. Okay, that means only one substrate can fit at a time. If suppose the substrate is there, it has finished the hypothesis and they have released themselves as a product. Only when this activity is now free, the other substrates and they attach to it. Okay,
Hmm?
you understood?
Yeah.
Okay, again a graphical representation. It will be this way, where here you have the rate of enzyme. Here you have the substrate concentration. Now, earlier when we observe everything reaches an optimum level, then comes down and they can be denatured, right? But
Mhm.
in this case, what do we observe here? Is it getting denatured?
No, this the rate of reaction like comes to a stop.
It comes
Not
to
to a stop. Like
it
there's only
becomes
so much
constant
rate.
value, right?
Mm-hmm.
Why? Because we saw this has reached a saturated level.
Yeah.
Okay, you understood? They have reached a saturated level, hence they become constant.
Yeah.
Clear so far what we have done?
Yeah.
Okay. Now, we saw factors which are affecting enzymes. We saw how enzymes work up. Now we'll see how enzymes and food are related to each other. Okay. The general biomolecules which we need inside our body. We need carbohydrates, we need proteins, we need lipids, okay, or fats. Now, these are the general biomolecules that are needed by the body. For what do we need them? We need them for our growth, for the repair of muscle tissues, and for metabolism. Right? Now, how do a body gets its biomolecules? Where do they get?
From food.
From food, right? So the food which we eat contain carbohydrates, proteins and lipids. These enter into the body through food. Now these molecules are very high molecules, right? They are huge, they are large molecules. So they need to be broken down so that they can pass from the intestine. The food we eat comes from the stomach to the intestine, right?
Uh-huh.
So from the intestine, it should go to the bloodstream. But these biomolecules are large ones. So what do we need to do is we need to break it down into smaller molecules so that they enter the bloodstream.
Yeah.
Okay. So now we are going to see how they are broken down. First one, we'll see how carbohydrates are broken down. Okay. Now carbohydrates, what do they have? We are talking about carbohydrates. We are talking about generally glucose, right? We have sucrose. We can also include starch, glycogen, etc. Okay, there are many carbohydrates which enter into our body through food. Yes, that
Wait, what does the second one say? Sucrose.
sucrose. Yes, sucrose.
Uh-huh.
What is sucrose?
A type of sugar.
A type of sugar. This is the basic table sugar which we use.
Yeah.
Okay. So these are the building blocks, the sorry, carbohydrates. What I was telling, we are talking about carbohydrates. Okay. Now the basic building block of a carbohydrate is referred to as a monosaccharide. Okay, monosaccharides first they will include the sugar, simple sugars like glucose, fructose, and all.
Uh-huh.
Many monosaccharides are formed, or many monosaccharides are joined together, they will form the disaccharide. when many disaccharides are joined together, what do we have is the polysaccharide. Okay, so we have simple sugars which come under monosaccharides. Then many monosaccharides form a disaccharide, like two monosaccharides form a disaccharide, and many disaccharides and monosaccharides together form the polysaccharides. Fine. Now we need to break this chain. So this is the heaviest part, right? We need to break down polysaccharide, then it will turn into disaccharide, then it will come into monosaccharide. So how are we going to break it? We have an enzyme as carbohydrates enzyme. Okay, this is going to break the disaccharide and the polysaccharides to my monosaccharides. You understood what is happening here?
Yeah.
Basically, my complex structure is my polysaccharide, right? Now I need to break this polysaccharide into the simplest form.
Wait, what's this enzyme?
Carbohydrates.
Yeah. Yeah.
These carbohydrates enzymes, they are produced in the mouth, or more
Yeah.
precisely in the saliva. Got it? Basically
Uh-huh.
present in the mouth. Some traces are also found in pancreas.
Yeah.
Pancreas also sometimes in small intestine. Clear with this carbohydrates part?
Sorry?
You're clear with how carbohydrates are breaking down?
Yeah.
Next, what we are going to see is about proteins. Okay, now proteins. Proteins are the final large molecule made from amino acids. So you got it? How proteins are made? Many amino acids they join together.
Yeah, yeah.
Okay, and they form proteins. Now, this protein in here we saw examples like glucose, sucrose, starch, and glycogen. Now, when we're talking about proteins, we have enzymes, right? We have hemoglobin, collagen. Also, we have traces of keratin. Okay, now these are proteins which are formed. Now, this protein we're talking about, they have hundreds, thousands of amino acids which are joined together, right? And now we need to break down this protein. So, what enzyme are we going to use? is we are going to use the enzyme protease. Okay,
Yeah.
we are using enzyme protease to break down the protein into smaller foods. You understood?
Yeah.
So once
Mm-hmm.
protein is broken down, what do they form? They form small molecules of amino acids.
Yeah.
You're clear with carbohydrates and proteins?
Yeah.
Okay. Next is lipids. Okay. Now these lipids, they include fats and oil together. Okay.
Yeah.
They form the fatty acids. Okay. They include the fatty acids and they also include the glycerol. What is a glycerol?
Um, is it a type of sugar too?
It is a carbohydrate, yes. Glycerol is nothing but a form of a carbohydrate only. Okay. And now you
Uh-huh.
need an enzyme to break this lipids. So what enzyme we are going to use is lipase. Okay. We are using an enzyme known as lipase enzyme, which is breaking down the proteins.
Yeah. I understand.
Understand this part?
Yeah.
So when these lipase are there, they break down these lipids into fatty acids or the glycerol. These lipids or the least lipase enzymes are produced in pancreas.
Mm-hmm.
Okay. You understood how they're all broken down?
Yeah. I understand
Okay.
this bit with enzymes.
Enzymes. Okay. Now tell me, do you have a subtopic which is related to enzymes and digestion or anything else related to enzymes?
There's this thing like enzyme cofactors and inhibitors. I'm not really sure about those.
Sorry, I couldn't hear you properly. Can you please repeat that again?
Cofactors and inhibitors.
Okay, inhibitors and cofactors. Okay, we'll do that. Okay, again, I will show you with the. Now there are two types of inhibitors. One, we have a competitive inhibitor and a non-competitive inhibitor. We'll see what are they. As you see, first one we'll talk about is competitive inhibitor. Okay, just have a look over here. Now this is
Yeah.
the enzyme. This is my substrate. This is my inhibitor. What is an inhibitor? The inhibitor binds to the enzyme. The chemical reaction which would be carried out by the enzyme does not happen. What is happening in my inhibitor? My action is not being carried out. Okay, this is inhibitors. Now what if there is a substrate which is no inhibition? Now if the substrate is going to bind to the active site, there should be some chemical reaction happening here, right?
Yeah.
Okay, and that is the reason why they have a competitive inhibitor. Both have the same, right? Both of them have the same shape. Now there is a competition among themselves as which one is going to go fast and fit into the enzyme. You're getting it? The substrate or the active site have a similar kind of a substrate. One which is a substrate, one which is an inhibitor. Now since both of them can go and fit into this active site, there is a competition between them as like who is going to go first and get fit. Okay.
Yeah.
That is known as a competitive inhibitor.
So an inhibitor just decreases the rate of reaction.
It decreases the rate of reaction. I mean, the enzyme activity doesn't happen here. Okay.
Mm-hmm.
Now, non-competitive. We saw here both of them have a similar structure and both have had the capability of going and attaching themselves to the active site. Okay. But here if you see, there is a substrate binding site and there is an inhibitor where you cannot fit that inhibitor into the enzyme. You're getting what is happening over here, non-competitive. The inhibitor as well as the substrate, they both look very different. That means only the substrate has a possibility of going and attaching itself to the active site, whereas the inhibitor is just binding with the enzyme. Okay, just a surface binding. It's just a kind of a surface binding which is happening here.
Yeah.
Okay, so now there is no competition as who's going to go there and release the end product. So naturally, what is happening? Uh, it has taken a substrate binding site. It is also giving an inhibitor to come into a way.
So it's just there, it doesn't actually have an effect.
No effect is happening.
Okay.
Okay, so there is no competition, so they termed as a non-competitive inhibitor. And here is the end product of the inhibitor. What do we see here as the end product? We have a substrate binding site. We have an a substrate which is binding to an enzyme. We have an inhibitor which is binding to an enzyme. The end product is released from them. That is, the inhibitor is being released out.
Yeah.
Now, what I want to do is just go through the entire thing which we have done on enzymes today. Ask me if you have any doubts, anywhere you have not understood.
No, I pretty much understand all of it.
Okay, good.
I was just recapping a bit.
Okay, just go through them. Just go through them.
Yeah. Oh, sorry. Could you quickly tell me what a cofactor is?
Cofactor.
Yeah, like an enzyme cofactor.
Okay, one moment. Now we saw that an enzyme is protein based, right? A cofactor is a non-protein chemical compound. Okay, cofactor is nothing but a non-protein chemical compound, or we can also mention them as a metallic ions. So now they need a metallic ion or a compound required for a enzyme's activity, right? Now these factors, they can be, yes, these cofactors can be considered as helper molecules.
Metallic ions or...
They are the metallic required for the enzyme activity.
Okay.
Okay, and as I said, they are also known as the helper molecules. What is happening is they are just assisting in this enzyme reactivity.
Yeah.
You understood what a cofactor is now?
Yeah.
Anything else?
I think that's most of it. Is there any other hypothesis for this?
For enzyme, we just one of them. You remember the lock and key?
Yeah, but is there another one?
Nothing specifically. Let me have a look at my book. No. For schooling level, few people have only lock and key hypothesis.
Okay.
that's it. Can we
Yeah.
conclude over here?
Sorry?
Can we conclude our class over here?
Yeah.
Fine.
Mm-hmm. Thank you.
Okay. Thank you.